Hello students, today we are going to be studying about the topic bacteriological determination of potability of water. The name of the module is Bacteriological Determination of Potability of Water under the unit Microbiology of Air, Soil and Water. I'm Ms. Lynette Fernandez, Associate Professor of PES's Ravi S. Naik College of Arts and Science, Farmagudi. Here we will be studying about the potable water and also standard bacteriological procedures carried out to determine potability of water. What we will be learning here is the definition of potable water and also the different techniques for bacteriological examination of domestic waters. What is potable water? Potable water is the one which is suitable for drinking and domestic use. It is free of disease producing microorganisms and of chemical substances which are deleterious to health. Such type of water is good for drinking. But sometimes when a large number of microorganisms and organic wastes and chemical substances are present in the water, that water is not good enough for drinking and it is referred to as non-potable water or contaminated water. How does the water get contaminated? The water gets contaminated with pathogenic microorganisms which are released into the water by intestinal discharges of human beings and men. This can be quite dangerous as it carries a lot of pathogenic organisms and they cause a lot of disease which are transmitted through water, such as typhoid fever, paratyphoid fever, amoebic dysentery, bacillary dysentery, cholera, etc. In the large intestine of human beings and animals, enterobacteria are the normal inhabitants, especially E. coli, which are designated collectively as coliforms, Streptococcus faecalis, and Clostridium perfringens. Presence of these bacteria in the water shows that fecal contamination has taken place in the water. What are the characteristics of intestinal bacteria, especially the coliforms? Why coliforms? Coliforms are found in large numbers. These coliforms are aerobic and they are facultatively anaerobic, gram-negative, and they are non-spore forming rod-shaped bacteria. They ferment lactose with acid and gas formation within 48 hours at 35 degrees C. Some of the examples of coliforms are E. coli, Enterobacter aerogens, Salmonella, and Klebsiella pneumoniae. There are standard bacteriological procedures which are used for determination of potability of water, which will tell whether the water has bacterial contamination or not, especially when the bacteria comes from the fecal matter. These two methods are standard multiple tube fermentation technique and the other one is membrane filtration technique. Standard multiple tube fermentation technique. Under this, there are three tests: presumptive test, confirmed test, and completed test. These tests are for coliform bacteria. Why coliform bacteria? Coliform bacteria, as we have said, are present in large numbers in the fecal matter, and so they can be easily identified. The most important organism in fecal coliforms is the E. coli. It is excreted in billions by an average person in a day. Generally, these E. coli live much longer in water than the other intestinal pathogens. So even if the other intestinal pathogens are no longer present in the water and if E. coli is detected, then we can say that fecal contamination has taken place because most of these E. coli comes from the excreta. So, E. coli are easily detected compared to the actual pathogens. In carrying out these bacteriological procedures, the collection of water sample is very important. It must be collected in a sterile bottle. It must be a representative of the supply from which it is taken. The bottle should not be contaminated at all, and the sample should show no contamination. Contamination of the sample must be avoided during and after sampling. And the sample must be tested as soon as possible after collection, otherwise, the bacteria might get killed even before they are tested. Multiple tube fermentation technique. There are three tests involved here: presumptive test, confirmed test, and completed test. And these tests are for identifying the fecal coliform. Presumptive test. In the presumptive test, there are a series of test tubes are taken, which are a series of test tubes containing single and double strength lactose broth or L'Oreal tryptose broth are used. These are inoculated with 10 ml, 1 ml, and 0.1 ml of water sample and incubated for 24 to 48 hours at 35 degrees C. After which, gas formation may take place in the Derham's, which will be observed in the Derham's tubes as placed at the bottom of the fermentation tubes. If gas formation takes place, then it signifies positive presumptive test. And if gas formation does not take place, then the test is negative. Next is the confirmed test. For carrying out a confirmed test, there are two methods which are used. In the first method, a drop of culture from the positive presumptive test is transferred to a brilliant green lactose bile broth, and this is incubated for 24 to 48 hours at 35 degrees C. If there is gas formation, then the confirmed test is positive, and if there is no gas formation, then the confirmed test is negative. The second method of confirmed test is a drop of culture from the positive presumptive test is taken to strict eosine methylene blue agar plates. Then these plates are incubated at 24 hours at 35 degrees C. Colonies will be formed, which may be of three types: typical colonies, atypical colonies, or normal colonies. If typical colonies are formed, this will show nucleated condition with or without a metallic sheen. Typical colonies show that the confirmed test is positive. Completed test. In the completed test, the most typical colonies of EMB plates are transferred to a lactose broth solution and incubated for 24 hours at 35 degrees C. If gas is produced, then positive completed test for coliforms, and if gas is not produced, then it is negative completed test for coliforms. Besides this, the most typical colonies from EMB plates have to be also strict on the agar slant for observing the bacteria developed there. These bacteria, if they are gram-negative, non-spore forming rods on agar slants, then it confirms that these bacteria are coliforms. Another type of reaction which is used for differentiating E. coli and E. aerogens, which are both coliforms, is Imvik reactions. These are a set of tests which are biochemical tests, and they help in differentiating between the two. E. coli and Enterobacter aerogens very closely resemble each other in morphology and cultural characteristics. But E. coli is a fecal contaminant and Enterobacter aerogens is a non-fecal contaminant. So these tests are important for differentiating between a fecal contaminant and a non-fecal contaminant. These are the different tests which are used, which will help in differentiating between E. coli and E. aerogens. Next is the membrane filtration technique. In the membrane filtration technique, there is a sterile filter disc which is used and it is placed in a filtration unit. A known volume of water sample is passed through this filter disc. The bacteria which are present in the water will get trapped in this filter disc. This filter disc membrane is then taken out aseptically and placed over a media. The medium supplies nutrients for the growth of the bacteria on the filter disc, and the bacteria starts growing on the filter disc, forming colonies, which can be studied later. With this, we finish off with the bacteriological determination of potability of water.